Yeah. Good evening, everyone, and welcome back to the channel. Today, we continue with monohybrid crosses. Now, in total, with monohybrid crosses, we have to cover 3 different types of dominance. We've already covered complete dominance, where in a heterozygous state, the dominant allele will determine the phenotype and the effects of the recessive allele are masked. But there are 2 more types of dominances that we have to cover, and that is incomplete dominance and codominance. Now, with incomplete dominance and codominance, here we focus on alleles that are both dominant. So with incomplete dominance and codominance, there are no recessive alleles, right? Let's try to make an example. Let's say we've got a black cow. Let's say we've got a black cow and we also have a white cow. Let's put it like this. So let's say the allele controlling black fur is going to be capital letter B. And let's say the allele controlling white fur is capital letter W. Now in this case, if this black cow was crossed with this white cow, they would produce an offspring with the genotype BW. But B is written as a capital letter, telling us that it is dominant. W is also written as a capital letter, telling us that it is dominant. If this was an example of incomplete dominance, this cow would be black and this one would be white. When we cross them, they are going to produce a third and an intermediate phenotype, right? So a black cow and a white cow would produce, for example, a cow that is gray. So the phenotype that is produced here is different to the phenotypes of the parent. So we call that incomplete dominance. We produce a third and a different phenotype. But the same can also apply to codominance. If this was an example of codominance, the same scenario would apply. We would have a black cow and we would also have a white cow. So this tells us that the genotype of the offspring would be capital letter B, capital letter W. But with codominance, the allele for black fur and the allele for white fur are said to be equally dominant. So when they're equally dominant in the phenotype, they're going to be expressed equally, right? So this offspring, the phenotype of the offspring will be a cow that is black and white. So the allele for black fur and the allele for white fur will be expressed equally in the phenotype. So that is the difference that exists between incomplete dominance and codominance. With incomplete dominance, we produce a third and a different phenotype. With codominance, we're going to produce we're going to produce a phenotype that is an equal expression of the allele for black and the allele for white, right? Now let's try to get into detail, starting with incomplete dominance. As we've already said, neither of the alleles is dominant over the other when we cover incomplete dominance, right? As such, since both alleles are written or expressed as capital letters, in a heterozygous condition, a third and a different phenotype is going to be produced. Let's make an example now. This is example 1. It says that the diagram below shows the inheritance of flower color in snapdragon plants. The 2 alleles controlling flower color are red. We'll note that the allele for red here is represented as a capital letter. And we know that once we see that the allele is represented by a capital letter, that is a dominant allele. The same also applies for white. White here is represented as capital letter W. And since it is written as a capital letter, it tells us that white is also dominant, right? So here we've got 2 alleles that are both dominant. Let's look at the example. It says that a red flower is crossed with a white flower. But when this red flower is crossed with this white flower, they produce a pink flower. What happened? So a flower that is red, in this sense, it only makes sense that the red flower has to have the genotype capital letter R, capital letter R. And the white flower has the genotype capital letter W, capital letter W. This is the only way that this red flower is going to be red, and this is the only way that this white flower is going to be white. But let's look at the offspring. We know that the genotype of the offspring, it is represented by two alleles, one coming from the paternal origin and the other coming from the maternal origin, which tells me that this pink flower received a capital letter R from the red flower and a capital letter W from the white flower. So this tells me that this is a type of incomplete dominance. Since two different phenotypes, when they are crossed with each other, they produced a third and a different phenotype. So this is an example of incomplete dominance. If it becomes RW, then it will be pink because neither of the alleles is going to be dominant over the other. So neither is going to be expressed and neither is going to be masked. As such, we produce a third and a different phenotype. The flowers can only be red if the genotype is capital letter R, capital letter R. They can only be white if the genotype is capital letter W, capital letter W. Let's try to answer the questions. 2.4.1, state the type of dominance shown by the snapdragon plant. As we've already said, this is incomplete. This is incomplete dominance. And the reason we're saying it is incomplete again, two different phenotypes are crossed and they produce a third and a different phenotype. 2.4.2, I guess that is the answer that I just gave. So it is simply because when the red flower is crossed with the white flower, neither of the alleles for red flower or white flower is dominant over the other, and as such, they produce an intermediate third phenotype. 2.4.3, a gardener crossed two pink-flowered snapdragon plants. Use a genetic cross to show the ratio of the expected phenotypes in the offspring. So, a gardener here used two pink flowers. So, it is a pink flower that is crossed with another pink flower. From our example here, we know that if it is capital letter R, it is going to be a red flower. If it is capital letter W, then it is going to be a white flower. As such, capital letter R, capital letter W will give us a pink flower. This tells us that the genotypes of the parents that are crossed here is capital letter R, capital letter W, capital letter R, capital letter W. Let's perform the cross. Now, we know that starting off in the far left, we've got P1. Those are parents of the first generation. In front of P1, we write phenotype. Now, what is a phenotype? Phenotype, that is the physical appearance of these organisms, and they did tell us the phenotype. It is that we crossed a pink flower, and another pink flower. So we crossed two pink flowers, right? Below the genotype, so below the phenotype comes genotype. And we know that the genotype for pink flower, it will be capital letter R, capital letter W, crossed with capital letter R, capital letter W. Below genotypes comes meiosis. And in front of meiosis, we write nothing. Below meiosis, we come to gametes. And the way we write gametes, we simply separate the genotype by a comma. So it will be capital letter R, comma, capital letter W, cross, capital letter R, comma, capital letter W. Below gametes, we come to fertilization. And we are going to use our Punnett square. So that is 3 boxes to the side, 3 boxes downwards. So it is 1, 2, 3, 1, 2, 3. In the top left box, we simply write gametes. And then in the 2 boxes ahead of gametes, we're going to take the gametes of the first pink flower. So we're going to have capital letter R here, capital letter W there. And below the word gametes, we're going to take the gametes of the second parent. So it is also going to be capital letter R, capital letter W. Now in this box, we're going to have capital letter R, capital letter R. In this box, we're going to have capital letter R, capital letter W. In this one, we're going to have capital letter R, capital letter W. In this one, we're going to have capital letter W, capital letter W. Now it is important to maintain structure here, so it will not be acceptable to write WR and in other instances you write RW. You have to be consistent. So RW in this case. Below P1 we're going to write F1, and in front of F1 we write genotype. And we simply copy the genotypes of the offspring. So it is going to be capital letter R, capital letter R, capital letter R, capital letter W, capital letter R, capital letter W, capital letter W, capital letter W. Below genotype now comes phenotype. So let's look at the phenotype. This flower is red, and we've got RW, these two are pink, and WW give us a white flower. So the phenotype is going to be one red flower, two pink flowers, and one white flower. But here we've not answered the question. The question was specific in that it wanted the phenotypic ratio. And let's do that. So we've got one red flower, so I'm going to say 1:2. We've got two pink flowers, is to 2, and we have one white flower, is to 1. So this is our final answer. It is 1:2:1. Since this cannot be simplified any further, this is our answer. It is important to note that when we write the phenotypic ratio or the genotypic ratio, a ratio has to be written in simplest form at all times. And we cannot simplify 1:2:1 any further, so that is our final answer. Let's perform another example. So this is example 2. It says that 2 alleles determine the texture of human hair. An allele that results in curly hair, so to have curly hair, we know it is capital letter R. And an allele that results in straight hair, to have straight hair, that is capital letter T. And they say that in a heterozygous condition, the hair texture will be wavy. Let's analyze this statement. So curly hair is controlled by capital letter R, and straight hair is controlled by capital letter T. And they are saying that in a heterozygous condition, so if this is a heterozygous condition, it is going to be RT, right? So this is a genotype consisting of an allele controlling curly hair and an allele controlling straight hair. But when these two alleles together make up the genotype, they are saying that they produce a third and a different phenotype, which is hair that is wavy. And we know that when two different phenotypes produce a third and an intermediate phenotype, that is an example of incomplete dominance. So the type of dominance here that we're focusing on is incomplete, simply because you are going to produce a third and a different phenotype. Let's try to answer the questions. 2.6.1, what type of dominance? Incomplete. How many genes control the texture of human hair? The question asking for how many genes control a particular trait, the answer is always 1. 2.6.3, Mr. Anderson has curly hair and Mrs. Anderson has wavy hair. Use a genetic cross to show the percentage of possible phenotypes of Mr. and Mrs. Anderson's children. So Mr. Anderson has got curly hair and Mrs. Anderson has wavy hair. Now we know that the allele for wavy hair is capital letter R. So Mr. Anderson's genotype has to be capital letter R, capital letter R. Because if we write this in a heterozygous condition, then this hair is no longer going to be curly. It will be wavy. And then Mrs. Anderson has got wavy hair. And we know that wavy hair is the third and the intermediate phenotype that is produced. As such, the genotype for Mrs. Anderson will be RT, which are capital letters, right? So let's try and perform this genetic cross. Again, here we're going to simply write P1 in front of P1. What follows? It is the phenotype. What follows is the phenotype. Let's go and copy again. Mr. Anderson, curly hair and wavy hair. So we're going to write curly hair, it is crossed with hair that is wavy. So curly hair is crossed with wavy hair. Below phenotype now comes genotype. Let's copy one more time. It is here we're going to have capital letter R, capital letter R, capital letter R, capital letter T. Let's continue. Below genotype, we find meiosis. And in front of meiosis, we write nothing. So below meiosis now comes gametes. With gametes, we simply separate the genotype by a comma. So it is going to be capital letter R, comma, capital letter R, crossed with capital letter R, comma, capital letter T. Below gametes now comes fertilization. Let's draw our Punnett square. So it is going to be 3 boxes sideways, 3 boxes downwards. So 1, 2, 3, 1, 2, 3. Top left, we write gametes. And then in front of gametes, we include the gametes for the parent with curly hair. So it is going to be capital letter R, capital letter R. And below gametes, we write the gametes of the parent with wavy hair. So it is going to be capital letter R, capital letter T. So in this box, it is going to be capital letter R, capital letter R. In this one, capital letter R, capital letter R. In this one, capital letter R, capital letter T. In this one, capital letter R, capital letter T. Below fertilization now comes F1. In front of F1, we simply write genotype. And we simply copy the genotype of the offspring. As such, it will be capital letter R, 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 capital letter T, capital letter R, capital letter T. Below genotype now comes phenotype. Now, we've got 2 offspring here that were produced, and they've got the genotype capital letter R, capital letter R. And we know that when you've got capital letter R, this is going to be curly. So there are 2 curly hair. And we know that when you've got capital letter R, capital letter T, you're going to have wavy hair. So here it is going to be 2 wavy hair. And that is our question answered. Let's continue. Now let's come to codominance. Now, with codominance, the alleles that we use are also going to be dominant. That is to say, they're all going to be capital letters. But in this sense, we say that the alleles are equally dominant, whereas in incomplete dominance, we say that neither of the alleles is dominant over the other. But with codominance, the alleles here that we're going to use, they're going to be equally dominant. And when they're equally dominant, they're going to be equally expressed in the phenotype. To go back to our starting example, if we had a black cow and a white cow, with the black fur being controlled by capital letter B and white fur being controlled by capital letter W, an offspring with the genotype capital letter B, capital letter W, would be black and white, simply because the alleles for black fur and the alleles for white fur are equally dominant. And when they're equally dominant, they become expressed equally in the phenotype. So this is codominance. We're also going to focus on codominance when we focus on blood groups, right? Let's perform an example involving codominance. Here it says that feather color in chickens is controlled by 2 alleles. We can have black fur and white feathers. A homozygous white feathered chicken. So we know that in order for this chicken to be white, it must have capital letter W. And if it is homozygous, it tells us that this white chicken is capital letter W, right? So a homozygous white chicken can be crossed with a homozygous black feathered chicken. And this chicken here is black. And we know that in order for this chicken to be black, it must have capital letter B. So the genotype for this chicken is going to be capital letter B, right? To produce speckled offspring. So this is this chicken here. It has got speckled feathers, as shown in the diagram below. So let's come up with the genotype for this speckled chicken. So since we know that the genotype is expressed by two alleles and one allele is coming from each parent respectively, it tells us that the genotype for this speckled chicken can only be capital letter B, capital letter W. The capital letter B coming from the black chicken, the capital letter W coming from the white chicken. Right? So these speckled feathers, we see that black feathers are equally expressed with white feathers. As such, this is a type of codominance. Right? So state the type of inheritance described. Codominance. Codominance. Right? Explain your answer. The alleles for black feathers and the alleles for white feathers are equally expressed in the phenotype. That is because we produce a speckled feather which has got black and white colors. So that is codominance. 2.3.3. If two speckled offspring were interbred, use a genetic cross to determine the genotypes and phenotypic ratio of their offspring. So here we're performing a genetic cross and we're using two speckled offspring. We're using two speckled offspring. So if they are both speckled feathers, it tells us that their genotype is capital letter B, capital letter W, capital letter B, capital letter W. But there's a very important thing to note here. What we are crossing now, it is the offspring. So it is the offspring of these two chickens, of chicken 1 and chicken 2. So these offspring now are going to act as parents. But we cannot say that these two speckled offspring are parents of the first generation because we already have the parents of the first generation here in the diagram. So these offspring are going to be parents of the second generation. As such, when we write our P1 here, it won't be P1, it will be P2 because the children are now becoming parents, right? So it is going to be parents of the second generation. Parents of the first generation was the black chicken and the white chicken. So if we're crossing those ones, we would have used P1. But because we're crossing the children of the black and the white chicken, P now has to become P2, right? I hope that makes sense. So in front of P2, the structure remains the same. So we're going to write phenotype. Going to write phenotype. They said it is speckled feathers. Speckled feathers. Speckled feathers crossed with another speckled feathers. Crossed with another speckled feathers. Right? Below phenotype comes genotype. And we know that to produce speckled feather, the genotype has to be heterozygous because capital letter B will give us black. Capital letter W will give us white. So in order to have black and white, which is speckled feathers, our genotype has to be capital letter B, capital letter W, crossed with capital letter B, capital letter W. Below genotypes now comes meiosis. We know that in front of meiosis, we write nothing. So let's continue. Below meiosis now come gametes. Now comes gametes. To write the gametes, we simply separate the genotype by a comma. So it will be capital letter B, comma, capital letter W, crossed with capital letter B, comma, capital letter W. Below gametes, we follow with fertilization. And to show fertilization, we draw our Punnett square. That is 3 boxes to the side, 3 boxes downwards. Top left, we write gametes. And in front of gametes, we simply write the gametes of parent number 1. So it is going to be capital letter B, capital letter W. Below gametes, we write the gametes of parent number 2. So it is going to be capital letter B, capital letter W. In the first box, it is B and B, so it will be capital letter B, capital letter B. In this box, it is going to be B and W, so it will be B, W. Here it is going to be W and B, but we are starting with B, W. Here it is going to be W, W, so it is going to be W, W, right? So below fertilization now comes F2. These are offspring of the second generation. These are offspring of the speckled feathers, which are children or offspring of the black and the white chicken, which were of the first generation. So in front of F2, we write gametes, right? Gametes. So let's simply copy our gametes there. So it will be capital letter B, capital letter B, capital letter B, capital letter W, capital letter B, capital letter W, capital letter W, capital letter W. Below gametes, we come to phenotype. So let's look. We've got capital letter B, capital letter B. This chicken is black. Capital letter B, capital letter W. In a heterozygous condition, we know that the allele for black and the allele for W, they are equally dominant. So when they're equally dominant, they're equally expressed in the phenotype as what we call speckled feathers. So capital letter B, capital letter W will give us speckled feathers. The same applies for this one. And capital letter W will give us a white chicken. So our phenotype here, we've got one with black feathers. One with black feathers. And then we're going to have two with speckled feathers. We're going to have two with speckled feathers. And finally, we're going to have one with white feathers. And then we're going to have one with white feathers. But the question was asking us to show the phenotypic ratio. So let's write it here on the side. Phenotypic ratio. So our phenotypic ratio, we've got one black feather, so it is going to be 1:2. Two speckled feathers, so 1:2, and one white feather, so our phenotypic ratio is going to be 1:2:1, and that is the simplest form of this ratio, and therefore we have answered our question. Let's perform a final example. Example 2. In some chickens, feather color is controlled by two alleles. We've got white feathers and we also have black feathers. Note that white feathers is controlled by capital letter W and black feathers is controlled by capital letter B. So they are both dominant, right? Let's read further. They say that in a homozygous white chicken, so if a homozygous white chicken is crossed with a homozygous black chicken, the offspring have feathers that are both black and white. Let's focus on this statement first. So a chicken that is homozygous for white and another chicken that is homozygous for black, when we cross them, they produce a chicken that is black and white. So this is a type of codominance, because we know that with codominance, neither of the alleles are dominant over the other. They are equally dominant. So when they're equally dominant, they become equally expressed in the phenotype, right? So that is why we are saying that we are going to produce a chicken that is both black and white. So this is a type of codominance, right? And they're saying that this chicken with black and white feathers is known as eminette. So a black chicken and a white chicken will give us an eminette chicken. That is a chicken that is black and white. So explain why this inheritance is an example of codominance. We're going to say that the allele for black feathers and the allele for white feathers are equally dominant. And when they're crossed, they're going to be equally expressed in the phenotype, right? So they're equally dominant. When you cross them, they become expressed equally in the phenotype. 2.4.2. The inheritance of which blood groups in humans show the same type of dominance as explained in question 2.4.1. So we actually see codominance in blood group AB. We're going to cover this later. We're going to show codominance with blood group AB, right? 2.4.3, a white chicken is crossed with an eminette chicken. Use a genetic cross to show all the possible phenotypes of the offspring. So if this chicken is white, the only possible genotype here is going to be capital letter W, capital letter W. If it is eminette, which is a combination of black and white, the only possible genotype will be capital letter B, capital letter W, right? So let's perform this genetic cross. So it is white and an eminette chicken. So it is going to be P1. In front of P1, it is going to be phenotype. So it is going to be a white chicken. That is going to be crossed with an What is the spelling again? Eminet. Eminet. So a white chicken there is crossed with an Eminet chicken. Below phenotype now comes genotype. Now comes genotype. White chicken, we know that the genotype can only be capital letter W, capital letter W. Eminet chicken, the genotype can only be capital letter B, capital letter W. Below genotypes comes meiosis. In front of meiosis, we write absolutely nothing. Below meiosis now comes gametes. Now comes gametes. To write gametes is very simple. We simply separate the genotype by a comma. So here it is also going to be capital letter B, capital letter W. Below gametes now comes fertilization. Now comes fertilization. And to show fertilization, it is simply 3 boxes sideways and the 3 boxes down. And this is our Punnett square. Top left, we write gametes again. In front of the word gametes in the 2 boxes, we're going to write the gametes of the first parent, which is the white chicken. So it is going to be capital letter W here and the second capital letter W there. Below gametes, we're going to write capital letter B, capital letter W. Those are the gametes of the second parent. So capital letter B, capital letter W. In this box, it is going to be capital letter B, capital letter W. In this box, capital letter B, capital letter W. In this box, capital letter W, capital letter W. In this box, capital letter W, capital letter W. So those are the genotypes that we're going to produce. So below fertilization now comes F1. These are offspring of the first generation. In front of F1, we write genotype. We write genotype. In front of genotype, we simply copy the genotype of the offspring that we produce there. So it is going to be capital letter B, capital letter W, capital letter B, capital letter W, capital letter W, capital letter W, capital letter W, capital letter W. Below genotype, we come to phenotype. So let's look at the first chicken. It is capital letter B, capital letter W, which means it is Eminet. The same applies for the second genotype. But the third and the fourth genotype are capital letter W, capital letter W respectively. So this is going to be a white chicken. So here our phenotype is that we're going to have 2 Eminet. We're going to have 2 Eminet and 2 white. 2 white. So that is our phenotype. We're going to have 2 chickens with 2 Eminet feathers and 2 white feathers. And that is our answer. If you like what we've had to say, please consider liking and subscribing to the channel. Until next time, bye-bye.